this week though.
Blood and heart review. So if you look at your schedule, you will have a quiz over this entire lab next Tuesday, okay? But I'm just going to review the blood stuff today, and then on Thursday I'll review the heart stuff. So here's the blood stuff. It's not too terrible. Make sure you can identify all this stuff under the microscope. I'll help you with that. I mean, I'm going to give you guys like good examples. I'm not going to pick like equivocal cells that look like they can be one thing. They're going to be like cream of the crop, because I don't want to argue with you guys. I just want to live my life, sail through. I don't want to engage in fights with anyone. I'm too old for that. So it's going to be perfectly crystal clear, and if you don't get it right, well, that's on you. All right. Sickle cell anemia, I'll show you that. Did anyone look at the sickle cell slide? I can't remember. I don't like our sickle cell slides because honestly I don't see a lot of sickling on them. So I don't know if you guys found anything, but I'll show you a good sickle cell slide. And then we'll talk about the blood typing results in a moment. Are we having a chair malfunction? Be careful. I don't want you to fall on your butt. This is the stuff you have to look forward to on Thursday. Don't stress about it now. Okay? You can stress about it on Thursday. Don't stress about it. All right, so here's a blood smear. Honestly, this is kind of far away. I wouldn't probably show you this low magnification for identifying the white blood cells. I mean, although you still can kind of see them. Can everybody tell what that is?
Is that a cylinder?
That's a neutrophil. There are like 4 little lobes to it. That's also like a neutrophil. There's also 4 lobes to it, but I'd probably go closer. Yeah, no, I will. And then that's going to be like probably a lymphocyte, but this is a little far away to really see. Here's a little closer still. What are these guys? They're all neutrophils. All right. If you see a crazy nucleus, it's probably a neutrophil. All right, with lots of little parts to it, it's a neutrophil. All the rest of these guys are red blood cells, except what do you suppose all those little tiny things are?
Platelets?
Platelets, right? They're kind of squeezing their way in there. Here's still closer magnification. This is a really good view. What are all these cells? All the white blood cells are? They're all neutrophils. Again, 60 to 70% of your white blood cells are neutrophils, so it shouldn't be shocking if you see a bunch of them, okay? What's this? More neutrophils, right? You can see, each one of these is considered one nucleus, but it's divided into these lobes by like little connecting things that are kind of thin, right? You can see the platelets are the small guys, obviously. What do you suppose this is? Anyone guess? That's an eosinophil, all right? Notice that the cytoplasm is kind of pinkish or reddish, okay? I mean, ideally, it'd be nice if you could really see chunky red dots, but at least you can kind of compare it and look sort of reddish. And then you've got essentially these 2 lobes. Now it's a little hard to see the connector piece, but you can kind of infer that there would be a connecting piece between the 2 of them. And that's that, what they describe as sort of the earmuff or the headphone-shaped nucleus. Okay, so that's an eosinophil. This is another eosinophil. Again, I think you can see the chunky red pink dots a little bit better here, and you can see the 2 lobes of the nucleus. What's this? The elusive basophil, right? Lots of chunky, big purple dots, no real nucleus. I mean, it's not a real sharp edge on the outer edge. That's another tip-off. It's very, honestly, I'd probably have to sit down for, I don't know, maybe at least 20 minutes and really focus. And by that time, I'd get so dizzy from moving the thing around that I'd be like, screw this. Here's another basophil, right? Not that we've ever seen one in real life. That's another basophil. What would these be? These are all lymphocytes. They're nice and round. You can, if you're lucky, like on these ones, see a little bit of cytoplasm. But, when you can't see the cytoplasm and all you see is the nucleus, that's when you can potentially mistake it for a basophil, right? What's this? It's another lymphocyte, big purple round nucleus. By the way, in the lymphatic system chapter, just to jump ahead real quick, we're going to be looking at things like the spleen and lymph nodes and the thymus. And all of those places have lots and lots of lymphocytes grouped together. And if you remember what one lymphocyte looks like, when you have a whole bunch of lymphocytes together, they look like these big purple areas because lymphocytes have this big purple nucleus. So just keep that in mind when we start looking at those things. When you see collections of purple, it usually means you got a bunch of lymphocytes kind of hanging out there. What's this? That's a monocyte. First of all, it's huge. And then second of all, you've got this kind of bent, horseshoe shape to it. Sometimes it looks a little bit more like, a kidney shape, a little bit, okay? So that's also a monocyte. This is just a summary slide showing you all 5 of them together. So, ingrain that in your mind. And then what's this from? Yeah, sickle cell. Notice that not all the cells are sickled, right? You got some that look relatively normal. Sickling only happens, you don't really need to know this for lab stuff, but you will need to know it for lecture. Sickling only really happens after the red blood cell has given up its oxygen. And depending on what you inherited, whether you have sickle cell disease or sickle cell trait, you also may not see as much sickling, right? It just really depends on how severe your particular case is, right? This is just a picture basically of that same kind of fake blood thing that we had back there, and just some practice things here. So what would you call this blood type?
A positive.
Yeah, this would be A positive. There's clumping or agglutination, whatever that's the fancy word for clumping. There's agglutination in the A spot, there's agglutination in the RH spot, but there's no agglutination in the B spot, so that's what gives you the A positive. What would this be? B negative, right? B because there's clumping here, and then the word negative because you don't see any clumping in the RH spot. And then what would this be?
AB negative.
Yeah, AB negative. This is basically the exact same one that I showed you that I did on my own up there, right? All right. And then the rest of the stuff is going to be hard stuff that we'll talk about later. Any questions about any of the stuff we covered in lab today? All right. Good night. What you're doing is you start by taking the sample of the fake blood. Now this is just called sample 3. There's sample 1, 2, 3, and 4. So you can try the different ones to see what they look like. It used to be back in the day, yeah, the old ones were a little bit better. They'd be like Mr. Green, like they had people's names, like it felt like it was a real patient, but now they just call it sample, whatever. So take this, and you start by putting like 2 drops of this blood in all 3 spots, right?
That's what I was going to ask. Can I just try it?
Okay, so you got 2 drops of the blood in all 3 spots. Then, what you're going to do is you're going to mix it with these 3 different test chemicals, right? This one in blue, this is the anti-A test, so you want to make sure you put that in the A spot. This is the well, let me go to this one first. This is the anti-B one that's in yellow there, so that's going to go there. And then this is it's called anti-D. That I think that's just an older name for the RH. It's the same thing. So you basically got those 3 different things. And there are different toothpicks in here. Make sure that you use a different toothpick for each one of the mixes that you're going to do. And you just take each corresponding test or vial, and then you put 2 drops into the corresponding spot and mix it with the blood. So here's the anti-A. Do 2 drops of that. Here's the anti-B. I'll do 2 drops of that. Okay. And then here's the anti-RH, and I'll do 2 drops of that. Now what this is doing is each of these chemicals are testing for the presence of each of those antigens. So in this spot, you're going to be looking for whether the A antigen is present. Here you're going to be looking for whether the B antigen is present. And there you're going to be looking for whether the RH antigen is present. How do you know it's present? You know it's present if you see, quote unquote, clumping or agglutination. It just means that the test chemical I gave will interact if the antigen is there, and it'll cause it to kind of clump together, and you'll see what I mean by that. So after you put those things in, take a toothpick and just kind of gently swirl each one around a little bit. And you got to make sure you use 3 separate toothpicks or you'll mess things up. Now, sometimes it can be a little tough to see the clumping. I can see it on this one pretty easily, and I'll go in closer here in a second. But one thing I recommend doing to bring it out to see it a little bit better is if you can find something that has print on it, and then just kind of put it over the thing that has print, and see if you can read through it or just like look through it, and I think it makes it show up a little bit better. Sometimes it's obvious and you don't really need to do that, but let me give it, sorry, your thing's not bright enough. Let me get this up a bit. And then, let me go in a little closer. Okay. Now, if you can see well enough, there's some clumping inside here. You see that? Is that bright enough for you? There's clumping inside there. There's also clumping inside here, but there's no clumping over there, right? So what would you call this blood type as?
A negative.
What is it?
A negative.
Well, there's clumping here and there's clumping there, so it's not.
There is clumping.
Yeah.
No.
So clumping here, clumping there, no clumping there would be what? A. So A there, we at least know A is present. We at least know B is present because there's clumping there, but then there's no clumping over in the RH spot. So this is AB negative blood. Okay. What if you had seen, and on the first quiz, on the lab quiz next week. Wait, is it? Yeah, it is next week. I could show you like something like this, or I might just describe the situation and say, oh, you got clumping in such and such a spot, but not in such and such. What would the blood type be? Okay. The point isn't really to see how well you can identify what clumping is. It's to understand what it means when there is clumping. So I may just describe it to you possibly. What if you did this and you didn't see clumping in any of the spots? What would that be? O negative, right? Again, clumping means that the antigen is there, and when the RH antigen is there, you want to use the word positive. When the RH antigen is not there, you want to use the word negative. What if you did it and you saw clumping in only the B spot, but no place else? B negative, good. What if you saw clumping in only the A spot, but no place else? A negative. What if you didn't see clumping anywhere? O negative, right? What if you saw clumping in all 3 spots? AB positive, right? So everyone's kind of see how that plays out. So let me put this on the back wall for those of you guys that want to try this, so you can look at this. I think if you see it up close, you'll see, you can definitely pick up on the clumping a lot clearer when you can see it actually at this distance too. That's pretty clear, I think. So let me put this back here. You want to try some of the other types, knock yourself out there.